I thought we would do here is just take a peek at your macromolecules lab here. When you're on your lesson page and you click in, you'll notice that this is a page that you come to, and you'll see we're not doing that top lab there. Okay, so that's just if somebody wanted to use that one. We're doing the one down here that's called the carbohydrates, proteins, lipids, and everyday foods. Now you'll notice that there are different categories here, like there's carbohydrates, and you'll see that some test tubes come up here that contain different things like glucose or potato juice. Proteins, it looks like the test tubes come up with, pre-selected with water, chicken soup, diet Sprite, egg white. Lipids, see some other ones that come up, and then everyday foods. When you get to this part, that'll be the last one, you can see that there are 4 to pick from here. And I'm going to have you pick any 3 that you want, and you'll do the testing yourself. But first of all, you have to go through, and kind of become familiar with the carbohydrates and the proteins and the lipids. So remember, when you're going to the chart and you're testing water for the carbohydrates in it, or the glucose, remember you're only going to test the ones that are here for the carbohydrates. So under your column that has carbohydrates, you'll only drop down and either say yes on water or no on water, or yes, there's carbohydrates in onion juice, or no, there's not. Yes or no on potato juice, yes or no on diet Sprite. Now you'll notice up here that we're testing for sugars here. For this particular group here only, it's the only one that has 2 to pick for. You also test for starches. And let's see, what did they change here? It looks like the only thing they changed really was the glucose here. They changed it to starch if I'm testing for starch. The other items that we're testing in the test tubes look like they stay the same. So just to kind of get you started, if you start with the carbohydrates, make sure that you read this. And one way of testing for sugar is by using Benedict's solution. So down here it says testing for starch, you're going to use iodine. So let's go back. I don't want to start with starches, I want to start with the sugar. So we'll be using the Benedict's reagent or Benedict's solution. Now as you read through here, when you click this to add the Benedict's solution to each of your test tubes here, you'll notice that it says here the Benedict's reagent changes color after heating. So we have, if there's, if in the presence of reducing sugars. So after you add the Benedict's here, you do not go by the color here. Let's see if anything changes. We have a light blue, and light blue here. You have to put it in a hot water bath. You'll notice that the water and the diet Sprite remained the light blue, but we did have one that changed color here. If we would have just gone by the original color, we wouldn't have made a correct determination as to whether there is sugar in that glucose solution. Okay, because originally it was blue, which would have said no. So then after you do the sugars, you'll want to, you see if you want to even do it again, you can reset the experiment, add the Benedict's solution. Look at that, the glucose, you're testing for sugar. Obviously glucose would have it, right? So not till I heat it though do I actually get a change in color. Then remember that you'll go back to this. I've kind of given you some helpful information so you don't have to research it further. Pale blue, when you're using Benedict's solution, that pale blue shows no reducing sugar. Green shows a little. And if it's yellow, orange, brown, red, and leading up, the yellow to the orange to the brown to red, it shows progressively more sugar. Okay, so you can just either put yes or no. You don't have to, unless you want to put a side note as to something having just a little bit more or a lot more sugar in it. Just a plus for yes, there is sugar, or a negative sign for no, there is no sugar will be adequate. Okay, so you can reset that. Then after you do that, you want to go to, and I'm not going to do all of this, but then you go to the testing for starch. And down here it'll tell you that it's the iodine that you would drop into each of those test tubes to see if starch is present. And it says a color change from yellow or brown to a blue-black. That blue-black indicates that starch is present. Okay, so you'll want to do that. And you'll want to put that data in the appropriate place in your chart. Then you'll go to proteins. Okay, you only have 4 that you're going to test for proteins here. When you finish, then you go to lipids, and you'll see that there's 1, 2, 3, 4, only 5. Okay, so the rest underneath your column for lipids, if you're not testing for it, just put an NA. Same thing here. Under the protein column, if you're not testing for it, just put NA. But if you are testing for it, you'll either want to have a plus or a negative sign indicating either present or not present, okay, for the protein. So if you have any other questions, just let me know. Like I said, I just wanted to kind of take you through it. I think some people got a little confused coming in and seeing this and then kind of making it through. But once you look at each thing together here, I think it'll help a lot for you. So, but again, feel free to call me or to contact me if you need any help at all, okay? And you still need a little bit more explanation on this. So thanks for watching. Hope it helped.